Research Foundation member, and also grants from their International Association for Study of the Lung Cancer. She is the author of several, almost more than 60 excellent peer-reviewed publications in reputed journals. So there are many other things she is engaged in. So she will share. She is also member of the IASLC's Cancer Development and Fellowship Committee, where that means she has established herself as one of the important and the contributor or the researcher in the field of lung cancer. Earlier, she did many other cancer. I think what was this cancer? Did you did in your PhD? Yeah, yeah. Yeah. So, but then you have taken a line which is a most prevalent and very dangerous cancer. Rather, lung cancer means it is 3 to 4 months. So far, so many patients I have referred, they have all gone for 4 to 5 months. But only one patient, exceptional, our colleague, Pankaj Sharma, he was a cancer researcher. He was in the US. What is that school? He was in, and there is a famous, what is that two famous school? Cancer. Yes. Huh? No, no, not Anderson. This is the lab. It is lab called. This is a, huh? So these are two labs. What is that? Dev Mukherjee is there. You know Dev Mukherjee? Yeah. Mayo Clinic. Mayo Clinic. And so he spent several years there, and he's a cancer researcher. He had paper in Cell and other, and but he ultimately he was a smoker, chain smoker rather, and because he was not able to overcome that habit, in spite of the year you shifted to New York, that is the year 2014, I shifted from Delhi University to here. And that is the year is the foundation year, a month. In April 2014, I moved here and this institute was created. And the lone person, and we have now 17 faculties, they must have talked, and they are all selected very expert faculty. Their majority are Ramaling Goswami fellows, and rest are 5 to 10 years spent abroad, all mostly in US and other places. And we have out of that, we have at this moment 17 faculties, and 7 are professors, and 7 are associate professors, and 4 are assistant professors. So among which these are few assistant professors that met Priyanka and Silpi. She was my student in DU, and she joined here as a young scientist and women scientist, and then become faculty recently. And Dr. Manoj, he was in BHU, and his wife is Sika. So she is a women scientist. These are women scientists. So they are. So other faculty you have met, I think, and you all have met. Virinder Kumar not. Yeah. Just introduce me, please. Just. So though it is a Institute of Molecular Medicine Stem Cell Research, we have three mainly the discipline, that is the cancer, molecular molecular oncology or molecular biology of cancer, and the other one is the stem cell and tissue engineering, and the host pathogen interaction. So she is in the host pathogen interaction. Dr. Virender Kumar, he is also a Ramalinga Swami fellow and associate professor, and they are already they come as a fellows, join, and then later on they are absorbed as regular faculty. So C has become regular faculty. Pallavi has become regular faculty. You met Dr. Manoj. He is also regular faculty. So they are Ramaling Goswami fellows. And Virender is going to be also accommodated. He is ending this fellowship. He works on what I wanted to say. You can tell what you are working on. And he is in the host pathogen interaction. Uh, and then the steps are all in this. Correct. Okay, so we can start and then we can go ahead with the. We welcome you to the AMSR, Amity Institute. We call AMSR, Amity Institute of Molecular Medicine and Stem Cell Research. So today the population is less because of the fact there are at a time that admission going on for a first year, and that is a orientation goes on, and there are including everybody. There is a any function starts in the Indian way here. The Havan is organized. Yes. Yeah, Havan is organized. So any good thing or the session starts with that. And then we have our examination. We run the MSc molecular biology of cancer. And I forgot to you also introduce. He is a, yeah, so we have also molecular the AMSR. Then I am also looking after the Amity Institute of Genome Engineering. So there are only three people. One is in the Switzerland. He has gone for leave, and Lian, and he is Professor Manoj Kumar, and another is he is not here. He is from South. He is also Ramaling Goswami fellow. He recently have the Nature Chemical Biology paper. So they are mainly on the plant genome engineering. So he is what is his name? Timappa, Ramesh Timappa. So he has gone. His father is not okay. So he is from South. So, these are today. We have another course we run. We did a MSc molecular biology of cancer, molecular cancer biology, then stem cell and regenerative biology, and then MSc in stem cell and regenerative biology, and MSc in molecular medicine. Then we have BSc in biochemistry, and this year we have started the MSc, BSc in biomedical sciences. Then also we have a MLT BSc, and that is 4 years courses all. And we run a medical courses that is very attractive, and 100% more than 100% is placement, and there is no faculty available in that. That is IVF program with the Indian Fertility Society, collaboration with that. So it is a medical plus basic scientist involved in this, and it's a very successful course. It is a sixth year running, and seventh year admission is going on. The sixth year examination is going on today. 3 days examination, and then will be viva in the last day, and the eighth is the concluding session that the programs he was giving. The concluding session, there are around 25 to 30 students. This year it will be 30. Earlier it was 30 but reduced, but now it has gained again. So that is clinical embryology, the diploma, 1 year diploma in clinical embryology. They do the 4 days in a year, the classes here. And another course is called the clinical assisted reproductive technology, clinical ART. They are all clinical people, MD, MS, in the gynecology. And they are already, many are appointed already in the private or government, or they may be fresh. So they are mostly they are 100% are clinicians. Here clinical plus basic in clinical embryology. So these two courses are very highly successful, and the medical people, they are accredited clinics, they practice. So that is there in the, I think there is Calcutta also, there is clinic perhaps. So all these states, they have majority of the states, but Delhi NCR, Chandigarh or Punjab, you have all the clinics are there, and they go for training. And this MSc courses people, they do 1 year course here, theory courses. 1 year they do the practical in the clinical practical in the clinics. So accredited clinic by the accredited by the IFS. So this is what, and the other people, the clinic at ART, they do 4 days in a year, 4 times 4 days each. They will come for a theory class here. So we teach also, and the clinical people come from different places. They come and stay in their guest house, and they teach. And even students sometimes also come and stay, those who are outstation. So that's what is very successful courses. And fees is very high in that. So 3.5 lakhs for the semester. So thank you very much, and all this that you could make it convenient to come. So it is nice to see you after a long time. And being my student, you have done excellent progress. And let us see what you are at it in the at the research level. So let us.
Is it possible to turn down the light? Because I will have a lot of light. Well, good afternoon, everyone. Thank you, Dr. Das, for that very, very generous introduction. And my life is in a full circle today because I started my research journey with Dr. Das when I was a master's fellow. And I really developed the love for research while I was in his lab. That was the first time I held a pipette. So, and I never looked back actually. And I'm extremely privileged to be trained by you. And thank you so much for all your guidance and being a mentor. I remember when I was applying for my PhD, he wrote letters, everything. So I'm very, very thankful for you for all the support I've received over the years. And thank you for guiding me and developing the love for research that has carried me. I mean, now I do what I love, and thanks to you for that.
I just interrupt here. I forgot to tell. His family is closely known to me because his father is a surgeon. He is a cancer surgeon, oral surgeons, and other surgery also he does. So he does every year some meetings. That is the society. What is called?
World Science Congress.
World Science Congress. He was one time also the president of the medical sections in the young Science Congress in South. And he invited me for a lecture, and I went there. Also gave a lecture.
Yeah.
And but this World Science Congress, yeah, he organizes every year, and many a times. And once he organized in Bangladesh, I was ready to go, but then that was such a problem. That is now is the biggest news.
Yeah.
And that was that they said that was cancelled because of that in Dhaka University. They told that you can have this meeting, but you cannot go out of the campus of the Dhaka University to the city for any sightseeing or marketing. You cannot go. So how can people will go and attend the conference and they will not go for sightseeing? So that's how it was cancelled. We never went. So that's how. So I have a regular connection with him, and thank you very much.
Thank you. So today I'm going to talk about a few projects in my lab about the novel biology and therapeutic targets for lung cancer. These are some of my disclosures, none really relevant for this talk. So the Sun Lab in Mount Sinai is really a translational lung cancer research lab. We study several processes that are connected to lung cancer biology. So we study cellular reprogramming, cancer progression. We study the cell intrinsic effects of drugs. We study how a drug changes the immune microenvironment. But the goal really of my lab is to develop novel therapeutics. We want to do precision medicine. That's all we think about. That's all we want to do. But I'm a molecular biologist by training. So what we do is molecular biology. We are very much rooted into cell biology, molecular biology, but we have adapted to new technologies um, like CRISPR, mouse modeling, single cell. These are just tools that we use with the goal is how we can give the best therapy to the right patient. That's all we think about. And I wear another hat. I'm also the director of the Lung Cancer PDX program. And under that, we collect tissue and blood from patients and we make mouse models. So they can be tissue derived mouse models or blood derived mouse models. And they are used really as a surrogate for patient samples, which are really hard to get. So we do co-clinical trials in the lab with novel investigational drugs, and we try to bring that to the clinic. So I recognize that not all of you work on lung cancer, maybe, and mainly the students at the back. So I'll give you a very sneak peek about what lung cancer is, what are the major subtypes. So lung cancer is the leading cause of cancer-related mortality in the United States and worldwide. So it kills more people than the next three cancers combined. And lung cancer can be divided broadly into non-small cell lung cancer and small cell lung cancer. Non-small cell lung cancer is about 85% of lung cancer diagnosis, and 15% of lung cancer diagnosis is small cell lung cancer. And so these are the major subtypes: lung adeno, lung squamous, small cell, and large cell carcinoma. My lab studies all these subtypes, but I think for the interest of time, today I'm going to talk about two projects of the two major aggressive types of lung cancer, one in lung adenocarcinoma and the other in small cell lung cancer. So these are the two. So first, the project that I'm going to talk about is in KRAS STK11 KEAP1 co-mutant lung adenocarcinoma. Now, when we think about precision therapeutics, not just in lung, in all diseases that you might study, we think about mutations. How can we divide the cancer based on mutations? It can be KRAS mutation, EGFR mutation. We hear about P53, we hear about RB1. These are the major mutated genes in cancer. So in non-small cell lung cancer, the precision therapeutics has really progressed in leaps and bounds over the last 10 years. It has really progressed a lot. And I think the reason for that is we were able to find these driver mutations. These are gain of function mutations. And it is easier to have therapies that are targeted against gain of function mutations. So namely EGFR, KRAS, names you might come across in your textbooks that, these are the major drivers of these cancer. But even we have gone one step further. Not just the driver mutation, we have now identified co-mutations. So if you have a patient that comes with KRAS mutation, we no longer just think about the KRAS mutation. We think about what other mutations are there with KRAS. And the study that I'm going to talk about today are these two genes, STK11 and KEAP1, that are co-mutated in with the KRAS mutant tumors. So this was a study that was published during my time at MSKCC, Memorial Sloan Kettering Cancer Center. What the group found was that in KRAS mutant tumors, these tumors are very aggressive to begin with. But within the KRAS mutant tumor, there are two genes that are co-mutant, STK11 and KEAP1. And these patients who have these mutations have the worst prognosis. So we designed a mathematical algorithm called the OncoCast risk score. And I will not go into the detail, but what you can appreciate here is that anything that is on the right side of this curve, anything that is high, the patients do worse. So the higher OncoCast risk score means the patient do worse. So what we noticed was, along with KRAS, if the patients have these two genes, STK11 and KEAP1, they almost always fall in the high risk category for the OncoCast risk score. So they do worse. So what is STK11 and KEAP1? So these are very important tumor suppressor genes. So STK11 is a serine threonine kinase 11 that encodes for a protein called the liver kinase B1. It directly phosphorylates and activates a family of 14 AMP kinase, very important for cell proliferation, just cell survival, and also metabolism. And STK11 mutation is present in about 33% of lung adenocarcinoma. That's a huge percentage because hundreds and thousands of cases are detected every year of lung adenocarcinoma. What is KEAP1? So KEAP1, it encodes for this, it is the negative regulator of this NRF2 protein. And NRF2 activates the expression of 200 antioxidant response elements. And antioxidant response elements is really crucial for maintaining the metabolic balance in your body. And KEAP1 and KRAS co-mutant lung adenocarcinoma, we and others have shown that they are dependent on glutamine metabolism. And KEAP1 mutant patients are seen in about 17% of KRAS mutant tumors. So again, a very high percentage. So we actually looked into 1,235 metastatic lung adenocarcinoma cases. This is a Kaplan-Meier curve. Anything that is high is good. Anything that is low is bad. Basically, that's all you need to remember. So in 1,235 metastatic lung adenocarcinoma cases that were profiled with next generation sequencing, we found that 10% of STK11, 10% of these cases were STK11 and KEAP1. 46% of them were KRAS wild type, and 53% of them were KRAS mutant. But if you see this black line here, yes, this particular line here, these are the patients that have KRAS, STK11, and KEAP1 co-mutation, and these are the patients that do really badly. They have the worst prognosis. So our hypothesis was that STK11 and KEAP1 co-mutation actually promotes lung cancer progression, and through a synergistic axis that may or may not be KRAS dependent. So for that, we went back to the lab and do what we did. We chose two cell lines, one that was KRAS wild type and the other that was KRAS mutant. And we actually knocked out STK11 and KEAP1 by CRISPR-Cas9. So we created the variants, which has an STK11 knockout, the KEAP1 knockout, the double knockouts, which has both STK11 and KEAP1. And we functionally characterized them. We wanted to see what is the functional characteristics. Do they proliferate more? Do they invade more? We wanted to see that. We wanted to look at the unique gene expression programs. If you have either the single or the co-mutation of these genes, how does it change the gene expression program? What do you see? And finally, we also wanted to find novel therapies because that's what we do. So we did a druggable genome library CRISPR-Cas9 screen in these models to find novel pathways or novel therapies for this co-mutant subset. So a little bit about, this is just the QC part. So in the H358 KRAS co-mutant cell line, we were able to successfully knock down STK11 and KEAP1, as you can see here in Western blot. The LKB1 protein is lost and the KEAP1 protein is lost, and for both the cases. So what we noticed right off the bat, that when you take out STK11 and KEAP1, the cells start proliferating much faster. So they proliferate faster, they invade faster. So just by losing these two genes, they're becoming more tumorigenic and more aggressive. Then, of course, we did bulk RNA sequencing of these lines. So the ones that have single mutation versus the co-mutation. We had previously shown about glutamine metabolism, but in this study, what we noticed was that the second pathway came up, which is called the ferroptosis pathway. How many of you, I'm asking the students at the back, how many of you are familiar with the ferroptosis pathway? Apoptosis, all of you are familiar with that, right? Okay. So apoptosis is programmed cell death, right? You all know about the proteins that are involved, like BCL2, BAX. They are very well known. So ferroptosis was actually identified by Stock-Brentwell in 2012. It's a newer form of cell death. It is a non-apoptotic form of cell death that is mainly iron dependent. And how we measure ferroptosis is by measuring the lipid peroxidation in the cells. And I'll talk about the method that we do that. So this was the first time that we had identified that ferroptosis is actually a vulnerability in lung cancer. And the several genes that we... Is this... Okay, it's coming. So several genes that are associated with metabolism and ferroptosis came up to be upregulated in the co-mutant tumors. These included aldoketoreductases, glutathione pathway, glutamine peroxidase, and the XC antiporter. So these are all very intrinsically related to ferroptosis that were higher in the co-mutant tumors. So this is just a sneak peek of what ferroptosis is. As I mentioned, it's a death by lipid peroxidation of the cells, and we are noticing more and more that happening in cancer. And the two master regulators of ferroptosis is GPX4, this protein right here, and SLC711, this membrane protein right there. So what we wanted to see is that does these co-mutant tumors, are they truly resistant to ferroptosis induction? So for that, we inhibited GPX4. As I said, GPX4 is a master regulator of ferroptosis. So we inhibited GPX4 by RSL3, which is a drug. And what we noticed was that in the cells that lost both STK11 and KEAP1, they were very resistant to ferroptosis induction. So they were not dying through ferroptosis. So which we predicted as one of the pathways that these cancer is adopting to survive. So then we did an assay called the body PC11 assay. It's a flow cytometry based assay. Mainly we look at the GFP levels. But what this assay does is it measures the lipid peroxidation levels. And the higher the lipid peroxidation, the higher the ferroptosis levels in your cells. If at any point the students at the back, you don't understand anything, please feel free to stop me because this is for you. So what we noticed is that in the H358 cells, which are usually STK11 and KEAP1 wild type, when you inhibit GPX4, you're able to introduce or have higher levels of lipid peroxidation. But when you take away STK11 and KEAP1, they become resistant to ferroptosis induction. And even in the A549 cells, which has a de novo loss of STK11 and KEAP1, we knocked in CRISPR-Cas9 mediated knock-in of LKB1 and KEAP1. And we noticed that we were able to make these cells more sensitive to ferroptosis induction, as you can see here. The right means more. So what this slide is actually telling you, three things. First of all, the loss of 7 and KEAP1 actually makes these cells more resistant to cell death by ferroptosis. Second, these have less lipid peroxidation. And if you introduce either STK11 and KEAP1, you are able to make them more sensitive. So both these genes actually coordinate this function of evasion of ferroptosis. So then we did a druggable genome library CRISPR-Cas9 screen to look at novel targets. So this is, again, we went back to the RNA-seq data and we found the gene that was at the top upregulated in the cells that lost STK11 and KEAP1 was this gene called the steryl CoA desaturase, SCD1. This was the first indication. We discovered this gene in lung cancer and how that works in lung cancer. And what we noticed in the CRISPR-Cas9 screen when we looked at the depletion score or the genes that were highly depleted, so in both the cell lines, we found that SCD was a significantly depleted gene in both our lines, which means these cells actually needed SCD to do the cell survival. So which means SCD is an essential gene for them to evade ferroptosis. So SCD is right here, this red box. So what SCD does, it is a central regulator of fatty acid metabolism. It converts saturated fatty acid to monounsaturated fatty acid. It's needed for your cells to just survive. So what we went back into the patient data. So we looked at over 6,000 patient data that have either KRAS, STK11, KEAP1 mutations in several combinations. And what we noticed was that even in the patient data, in over 6,000 cases that we studied, SCD and SLC7A11, the two main regulators of ferroptosis evasion, were higher in the STK11 and KEAP1 co-mutant tumors. So we had our validation, not just in cell line, but also in the clinical cases. So then we knocked out SCD and we wanted to see, is SCD really crucial for the survival of these co-mutant tumors? So this is what we call a clonal competition assay. So in the clonal competition assay, what we noticed was that this is a busy slide, so I just want you to concentrate on what this the SG-SAFE and the SCD. So in the cells that had an intact STK11 and KEAP1, they did not really die that much with SCD depletion. But in the cells that already had an STK11 and KEAP1 knockout, we saw significant cell death in 12 days. So it went all the way from 85% of cell viability to 9.7% of cell viability. So the SCD was crucial for these co-mutant cells to survive. So then we wanted to see the mechanism of how SCD is actually working, because there were no reports of SCD in lung cancer before this. So what we noticed was that is working through the AKT GSK3 beta axis. I'm sure all of you have heard about it in your textbooks. It's a really crucial pathway for the cancer cell survival. So it is actually working through the AKT GSK3 beta axis. And what we noticed was that SCD inhibition actually decreases AKT levels, increases the levels of GSK3 beta. And through that axis, it leads to the upregulation of SLC711 and then lipid peroxidation. I have a schema at the end, but that is the axis through which SCD is actually working. So what then we did metabolomics. So we did something called the fatty acid methyl ester analysis, which looks at the fatty acid levels in your cell when you take out SCD versus when you don't. And we noticed that there was a family of 16 fatty acid families that were uniquely downregulated when you take out SCD in these co-mutant models. And we have investigated that. This is a work in progress, as you can see. This is now submitted for publication. But what we noticed was that the phosphatidylcholine and phosphatidylethanolamine families were the ones that SCD were controlling. So these were the fatty acid families that they were controlling. So, of course, then we wanted to see what happens in vivo. Can we do tumor regression in vivo? For that, we went to our mouse models. This is an immunocompromised mouse model, which we injected with our cell lines. And what we noticed was that if you take out SCD, which is this yellow line here, and the inhibitor is here, I don't know if you can see, it's really small, but this is an inhibitor of SCD, which is the A939572. So we did that either as a single agent or in combination with SLC7A11 inhibitor. And the red line, which is the combination strategy. So in a mouse study, the lower the line is, the better it is, because the tumor is regressing. So the ones that already had the co-mutant tumors, they were very highly growing. But when you inhibit either SCD or SLC7A11, you get decrease in tumor growth. But when we knocked in either LKB1 or KEAP1, we see that this tumor growth can be reversed. So it's actually working towards promoting this tumor growth. So for this part of my talk, I'm going to conclude the lung adeno portion of my talk. So STK11 KEAP1 co-mutation, what we found in our study is that it predicts the shortest overall survival in patients. And it promotes tumor proliferation in vitro and tumor growth in vivo. The tumor grows really fast. We discovered that it promotes higher expression of pheroptosis protected genes, and it's resistant to pheroptosis inducing agents. This is a novel pathway through which this cancer is actually escaping cell death. And inhibition of SCD prevents growth and sensitizes pheroptosis induction. The future studies is that how does SCD regulate survival in the co-mutant model? So we are going deeper into the mechanism. We have some indication, I mentioned through the metabolomics analysis, but we are going deeper. And how do we develop? So the inhibitors that we currently have, because we ultimately want to take it to patients, the inhibitors that we currently have are not that specific. They have a lot of off-target effects. So in order to take it to patients, we're actually designing better compounds with less cytotoxic effects. So we're doing SCD inhibitor construction right now, and we want to take it to patients very, very, very soon. So with that, I'll switch gears a little bit and talk about another cancer that I have studied for about almost 10 years now. And it's my favorite project. It's like my favorite baby. So small cell lung cancer. So small cell lung cancer is 15% of lung cancer diagnosis. It's a very small percentage, but it is the most aggressive form of lung cancer. The patients have very, very, very poor prognosis. They have months to survive. Like once you have a diagnosis of small cell lung cancer, it's really till now a death sentence to you because you have weeks to months to survive. So in the United States and also globally, it's actually one of the leading causes of cancer-related mortality. In the United States, it's the sixth most common cause. 30,000 people die every year of small, and globally about 250,000 worldwide. So very aggressive form of cancer. When I started during my postdoc studying this cancer, it was really slim pickings. We did not understand the biology. We did not understand the therapy that were there for small cell lung cancer. So it was really a bad state of the disease right now. It has progressed so much, work by us and others, of course. So when a patient with small cell lung cancer comes into the clinic as a frontline therapy, they're treated with chemotherapy and immunotherapy. So there are no targeted therapies available for these patients. They get chemo and they get anti-PD-L1. I think in India, they only get chemo at this point. So they will be treated with carboplatin or cisplatin along with etoposide. They initially respond for months, like it's a very short response rate. We get deep responses, about 60% objective response rate, but that response only sustains 3 to 4 months that the patient actually has that kind of regression. The disease comes back very, very quickly and almost always. And there are almost no therapies in the second line setting. What that leads to is a 5-year overall survival of less than 1% for the extensive stage disease. And about 70% of these patients come with extensive stage disease. So it's a really bad scenario for these patients. So when we look at the genetic makeup of small cell lung cancer, so these patients come with very long smoking history. They're usually between the age of 40 to 60, and they have about 20 years of smoking history behind them. So what that does is they have a very high genomic instability. If you look at their genome, it's very, very unstable. There are a lot of there are a lot of like intrachromosomal and interchromosomal rearrangements. There are a lot of mutations that they have. And this is just a circus plot that's showing that. And when you look at the mutations, when you just look at the heavily mutated genes, the major mutations that we see are in P53 and RB1, the two major tumor suppressors. The problem with that, with having tumor suppressor loss, any guesses? What is the problem with having a tumor suppressor loss in your cancer? It's very hard to drug it. There are really the tumor suppressors, since these are loss of function mutations, you can't really have a drug targeting a loss of function mutation and especially a tumor suppressor. So in any cancer that has this heavy percentage of tumor suppressor loss, it becomes virtually undruggable. So this is what we found in our small cell lung cancer data sets, is we could not drug this based on mutation. So then we actually looked into, so this is just showing you how badly we have done, like in terms of therapies. This is just a slide showing that we have failed. So for the longest time, chemotherapy was the only option. For 40 years, chemotherapy was the only option. The first positive clinical trial was in 2018, very, very recently, which is the Empower 133 trial, which led to the approval of PD-L1 in the frontline setting for small cell lung cancer. And I'll show you, even with the approval, how badly we are doing. The second approval was with Caspian study. So atezolizumab got approved with Empower 133. It's a PD-L1, anti-PD-L1 agent. And with the Caspian study, we had the second approval of durvalumab, which is another anti-PD-L1 agent. Everything else, negative trial. It failed. There are many other trials right now. We have Keynote 604, Capstone 1, Atrium 005, Skyscraper 2. So a lot of them are these adding immunotherapy regimens to chemotherapy, but most of them are actually failed trials. Now, again, I'll go back to the Kaplan-Meier curve, and I want to tell you how badly we are doing. I'm not really coming across well with these slides, but this is the state of the disease. So there are three major phase 3 trials that happen in the frontline therapy for small cell lung cancer: Empower 133, Caspian, and Keynote 604. And there are two major trials that happen in the second line setting for small cell lung cancer, which is Checkmate 032 and Keynote 028. But what you can notice right off the bat, the progression-free survival rule is 10%. Only 10% of the patients benefit from any trial, which means 90% of the patients have no benefit from any existing therapy. So even with the drugs that are approved, which is Empower 133 and Caspian, Atezo and Durva, there is so much room for improvement for these patients. So that's what we do in the lab. We want to raise the tail of the curve. We want more patients to survive, and we want them to have a durable survival, so years of survival. So if you think about the genomics of small cell lung cancer, you would think that they would respond better to immunotherapy, because any cancer that has a high mutation load, according to our theory, it has more neoantigens. So it will be more visible to the immune system. So theoretically, if you go back to your textbooks, they should respond to immunotherapy because the pros are high mutation burden, genomic instability, paraneoplastic syndrome. But the problem is there are many cons. This is an immunosuppressive cancer because it is inherently has a transcriptional loss of MHC class 1, which is an antigen presentation machinery. And it also has a less infiltration of cytotoxic T lymphocytes, which are the good T cells. And we have universal low expression of PD-L1. So the goal, one of the projects in the lab is how do we make these cold tumors hot? So how do we make this immunosuppressive microenvironment to an immunoresponsive microenvironment? So again, I'll just show you the slide that I showed you that these have a loss of function mutation in P53 and RB1, which means the cell cycle, it's cell cycle 101, that your G1S checkpoint is actually compromised if you have a P53 loss. So anyone at the back, what are the two major checkpoints in your cells? The G1S and the G2M, correct? This is what the cells actually rely upon if there are any damage to the cells. So if your G1S checkpoint is actually compromised, now the cell depends upon the G2M checkpoint for its any arrest upon DNA damage. So if you can take away the G2M kinases, either with drug or with genetics, you really have a potential for tumor killing. So either you take away repair of DNA with PARP inhibition, or you take out CHEK1, ATR, and V1, which are really the master regulators of the G2M kinases. And that leads to tumor cell killing. So we were the first to show that DNA damage response is actually a therapeutic target in small cell lung cancer. So we showed for the first time that CHEK1 is actually a therapeutic target in small cell. We went on to talk about V1. We showed that as a target. We also presented the mechanisms of resistance to these drugs that led to the first phase 1 trials of a PARP inhibitor in small cell lung cancer. And since then, there have been several trials that have been launched in the field, which is looking into either CHEK1, V1, ATR, or PARP, either as a single agent or in combination with chemotherapy in small cell lung cancer. So this is an entire field that now investigates DNA damage response in small cell lung cancer, including my lab. So interestingly, what my postdoc, this work was led by Hirogato Taniguchi. So what my postdoc noticed was that when we were treating our cell lines with either a CHEK1 inhibitor or an ATR inhibitor, we were getting structures like that. It's not very visible here. It's more visible in my computer. These are the structures which we called micronuclei. So what are micronuclei? So micronuclei are DNA-containing structures that are separate from the normal nucleus of a cell. And they are biomarkers. They actually tell you that there has been an aberrant chromosomal segregation. So these cells have undergone aberrant chromosomal segregations. That's why there is this DNA-containing structure which is separate from your nucleus. So we started noticing severe micronuclei formation when we were treating these cells with these DNA damage response agents. So then we predicted, and this is why you should read and talk about things that are outside your own field, because we went back to virus literature. Micronuclei formation has been very well studied in viral mimicry. So when you have micronuclei formation, that leads to the cytosolic DNA formation. So DNA gets released in the cytosol of your cells. What that does is it recruits this family of proteins called a cGAS family of proteins. And that recruits stimulator of the interferon genes. And this stimulator of interferon genes has been known from the virus literature to then lead to this complex with TBK1 IRF3 that leads to the type 1 interferon formation. So when we treated our cells with these DNA damage inhibitors, we found for the first time in small cell lung cancer that DNA damage can actually change the innate immune microenvironment in small cell lung cancer because it was activating the cGAS STING pathway. Every response in the cGAS STING pathway was actually getting upregulated. So we predicted that if we were able to upregulate or activate the cGAS STING pathway, which means that combining a DDR agent or a DNA damage response inhibitor with immunotherapy will actually work better. So for that, we went into our immunocompetent mouse model. These are genetically engineered mouse models, which has a conditional loss of P53, RB1, and P130. So we do an intratracheal intubation of HCMV Cree into the lung, and then we treat them with our drug of choice, either as a single agent or in combination with immunotherapy. So what we noticed was that single agent DNA ATR inhibitor actually works really well. If you can see here, the black line and the red line, these tumors are really aggressive. So you go from 100 cubic millimeter to 1500 or 2000 cubic millimeter in a matter of 3 weeks. This is how aggressive the tumor is. And anti-PD-L1 by itself does not work. But when you treat them with an ATR inhibitor, which is this blue line right here, it has significant delay in tumor growth. And with the combination with anti-PD-L1, we were getting tumor clearance. So the tumor was vanishing. And this is a very interesting observation for this aggressive tumor. So we resected some of these tumors and we did multicolor flow cytometry because we wanted to look at the immune subsets. And we found that upon treatment with either an ATR inhibitor by itself or in combination with immunotherapy, we were getting significant recruitment of the CD8 positive T lymphocytes, which are your good T cells, and also increase in the effector memory T cells. What effector memory T cells tells you is this response will be sustained. So then the question that we asked is that we are getting CD8 recruitment, but is CD8 really important for this anti-tumor response or not? So we depleted CD8 in our mouse models, and this is just with the IgG control. And when you deplete CD8, I really want you to concentrate just on this, this solid green line and the dotted green line. When you deplete CD8 in these models, you're able to reverse the anti-tumor response, which means CD8 is required for the anti-tumor response to happen. So then, since we were getting activation of the cGAS STING pathway, the next question we asked, is STING important? So to prove if anything is important, you really have to take it out of the equation. That's what you do in the cell culture and everything. So you have to take it out of the equation and then see if the response is still maintained. So we knocked out STING in our mouse model. And what we noticed that knocking out STING truly can reverse the anti-tumor response because the activation of the STING pathway is important for this DNA damage response inhibitors to actually have its anti-tumor effect. So then we wanted to see, okay, so we have done the mouse modeling. Where do we introduce that in patients? Do we introduce it in front line with the chemoimmunotherapy or in second line after they have responded or after they have been treated with chemoimmunotherapy? So we mimic that in our mouse model. So the mouse got 4 cycles of chemoimmunotherapy and then they were randomized to get either anti-PD-L1 maintenance or ceralasertib, which is an ATR inhibitor by itself, or the combination. And what we actually predicted, and the hypothesis actually came true, is that ATR inhibitor, even with pre-treated tumors, the tumors that have been pre-treated with chemotherapy and immunotherapy, they have a longer survival. They have a significant longer survival as a single agent ATR inhibitor or in combination with immunotherapy. So we're increasing the survival of patients even in the second line setting. So this was a very important observation. Another thing came up, which we were not expecting. And that's the importance. Like when you do your research, please don't throw away any data because the data will tell you something. Even if it's not something you expect, it's something the biology is telling you. So you need to follow that. So we noticed, and we weren't expecting this, we also were getting MHC class 1 upregulation. We had no idea that DDR would actually increase MHC class 1, but it was a very interesting observation for us. And we were getting it in our mouse model, we were getting it in our cell lines, we did flow cytometry, we did RNA sequencing, we did RT-PCR. In all levels, we were getting upregulation of MHC class 1 genes when we treated it with an ATR inhibitor. So we no longer could call it junk data because we were getting it in multiple platforms. So this was a phase 1-2 trial, which is a proof of concept trial looking into ATR inhibitor in berzosertib in combination with topotecan, which is a topoisomerase inhibitor. So we were lucky to collect pre-treated tumors and post-treated tumors. So the question we asked is that, great, we're seeing it in our mouse model. Is it a mouse model artifact? Or do we see MHC class 1 upregulation also in humans that have been treated with an ATR inhibitor? So when we did RNA sequencing, it came up. MHC class 1, MHC class 2, significantly upregulated in patient tumors that were treated with berzosertib in combination with topotecan. So this tells us that cGAS STING pathway, not just the innate immune pathway, but also antigen presentation can change with the DNA damage response inhibitor. And so I'll summarize this part. is that ATR inhibitor actually leads to the cGAS-STING pathway activation, and that launches a multimodal immune activation with the cGAS-STING pathway. It can increase HLA, beta 2 microglobulin. I'm not going into the mechanism for the interest of time, but we do see MHC class 1 upregulation. I also haven't spoken about DAMPs, which is immunological cell death markers, which we get. But I did speak about chemokines and cytokines, the type 1 interferons that gets upregulated. What that leads to is this tumor-associated macrophage repolarization, increased M1 macrophage, increased MHC class 1 and CD8 infiltration, which leads to the anti-tumor benefit that we get in our mouse model. So we have published significantly in this field, but what I think is the most gratifying thing is that we have launched clinical trials that are now combining ATR inhibitor, PARP inhibitor in combination with immunotherapy for small cell lung cancer, either in the frontline setting or in the second line setting. And we are very excited to see what that leads to in patients. So for the last few minutes of my talk, I'll talk about, this is a very new study that we are doing where we talk about drug resistance. So a major focus of the lab is not just finding novel targets and novel drugs, is to also investigate how does a drug become resistant? Because cancer, that's the problem. The drugs that we currently give patients, they have a very short window where they work until the patients don't respond to the drug. So for that, there's an example. We took the example of EGFR mutant lung adenocarcinoma, which is treated with tyrosine kinase inhibitor that targets the EGFR mutation. And usually the drugs that are there is osimertinib. But interestingly, what we started noticing was that these patients, about 14 to 20% of them, were actually completely changing histology. They were no longer remaining lung adenocarcinoma. They were changing to a completely different tumor type, which is small cell lung cancer. As you can see under the microscope, they look completely different. They look like two different tumors, but they have evolved from the same tumor. But this is, the more and more we started, studying about this phenomena, we noticed that this is not just restricted to EGFR. This happens in ALK rearranged tumors. This happens in immunotherapy. This happens in KRAS. And interestingly, this happens in other cancers, prostate cancer, pancreatic cancer, melanoma. They all become resistant by this process called lineage plasticity or histological transformation, where the cancer completely changes from one form to another form. And this is very hard because then the drugs no longer work. So what we identified were these 11 cases, which is mixed histology. It's very interesting because we wanted to study this in close proximity. So we took the histology slide and we were able to see lung adenocarcinoma and small cell in the same histological section. So we microdissected these samples from the histology slide we did whole exome sequencing from these histology sections. EPIC, which is a methylation array, because we wanted to look at the methylation levels of these genes. We did bulk RNA sequencing and immunohistochemistry. So when we looked at the mutation data, the genes that came out were P53 and RB1 loss. So if there is a lung adenocarcinoma tumor that is EGFR mutant, and they also have P53 and RB1 loss, and they're treated with tyrosine kinase inhibitor, they will most likely become resistant by transforming to a different type of tumor. That's what we predicted from this analysis. And when we compared it to TCGA, which we considered never transformed lung adenocarcinoma, we noticed that P53 and RB1 were the top predictors of these tumors that would transform. So EGFR can become resistant by several phenomena. It can develop a secondary mutation. It can turn on a feedback loop or other pathways. But if they have an EGFR mutation along with P53 and RB1 loss, histological transformation was the chosen mechanism of drug resistance. So when we looked into the RNA-seq data, interestingly, several pathways came up: cell cycle, DNA repair, PRC2 complex, Wnt signaling, and Notch inhibition, which were predictors of the histological transformation. And we were seeing decrease in the immune pathways. We were seeing decrease in innate immunity, cytokine signaling, neutrophil degranulation, T cell immunity. So immune suppression were one of the main predictors of drug resistance in these tumors. So we did multiplex ion beam imaging, which can look up to 40 colors. And what you notice is that the small cell section in this mixed histology section is actually an immune desert. So there is exclusion of MHC1, MHC2. It is truly excluding the immune microenvironment. So this is immunosuppression is one of the main markers. Then we wanted to see how do we treat these patients. This is what we do. So we wanted to see how do we treat these patients. So if you remember from the RNA-seq data, we were seeing several PI3K-AKT, Wnt-AKT signaling that were coming up, Wnt-beta-catenin signaling that were coming up. We treated with all the inhibitors, and the two main inhibitors that actually worked in our patient-derived xenograft models. And I'll walk you through the data. So we treated EGFR mutant PDX models that actually were taken from patients who ultimately developed small cell lung cancer. So they transformed in the patient, and we collected these tumors before they transformed. So as you can see here, osimertinib, which is a tyrosine kinase inhibitor, it doesn't really work in these PDXs. But when you combine them with the PI3K-AKT inhibitor, which is samatolisib, we were getting significant delay in tumor growth. And the second drug that we treated in our lab works a lot on lurbinectedin, is that lurbinectedin has a gangbuster effect when you treat these PDXs with lurbinectedin, even as a single agent, which is this blue line here, and the red line, which is a combination of osimertinib with lurbinectedin. It is working really well in the PDX model. So right now, we just started a trial where we are going to follow up these EGFR mutant patients who have P53 and RB1 loss. We are going to follow them up. When they develop resistance by transforming to small cell lung cancer, they are going to get osimertinib in combination with lurbinectedin. We're actually doing the trial right now. Finally, I'll go a little bit into the single cell data and I'll conclude there. So we wanted to see at the single cell level. So what I showed you is bulk RNA sequencing. But the problem with bulk RNA sequencing is that you only get snapshots of what's happening when the tumor was collected. And you miss the information of the immune microenvironment. So we did single cell RNA sequencing from a pre-transformed lung adeno and a transformed lung adeno tumor. And we also did it from a de novo small cell and transformed small cell. The question was very simple. What are the genes that predicts that a lung adenocarcinoma would transform? And how is a transformed small cell tumor different from a classical small cell tumor? The two genes came up is SOX2 and FOX2B, and we are investigating that in the lab right now. So then, in the single cell RNA sequencing, we wanted to look at the broader scale. How are these tumors different? So in a de novo small cell, as you can see, there's a lot of heterogeneity in the tumors. But when these tumors actually evolve from lung adenocarcinoma, the intertumoral heterogeneity really, really explodes. They're very, very different from each other. Can anyone tell me what's the problem with having a heterogeneous tumor? Therapy becomes a problem. Because now the tumors, every patient is different from each other. One therapy fits all will definitely not work. Now each patient has to be tailored. So precision medicine becomes absolutely important for these transformed tumors because they no longer look like the same tumor. Even when we look at the intratumoral heterogeneity within the same tumor, we found that these transformed tumors has huge intratumoral heterogeneity, which means within the same tumor, there were more than one driving transcription factors. So this is like you're adding complexity upon complexity, but that's really how complex a tumor is. And that's why we fail so many times in controlling tumors in our patients. So with that, this is really a concluding slide where we proposed this process of lineage plasticity in lung cancer. So lung cancer, which has an EGFR mutation, right now we are also looking into KRAS and ALK. When they have a P53 and RB1 loss, they change into these mixed histology tumors. They're changing to small cell lung cancer. And this is accompanied by several transcription factors that we have identified. There are a few transcription factors that I've mentioned here, like PAU3F2, 1CUT2, and FOXN4. And interestingly, these are the same transcription factor that came up even in prostate literature or pancreatic cancer literature. So there's a convergence of histology. So when they transform, they become really heterogeneous. There we see higher amount of intratumoral heterogeneity, intratumoral heterogeneity. There are many, many unanswered questions, but we really want to study how the immune evasion happens during this process of transformation. And of course, what are the therapies that can be targeted? These are some of the therapies that I've mentioned, but there are many more that we are actively investigating. We are studying what are the other mutations that can predict transformation. We are studying what are the other molecular drivers or transcription factors. And we are studying what decides how heterogeneous the tumor will become. So, conclusion, you should always adapt new tools if they are available to you. So, be bold, adapt new tools, study about new tools. CRISPR-Cas9 and single cell profiling. So the new tools in the toolbox can really help you define novel targets and novel biology. So comprehensive expression profiling, single cell sequencing, and CRISPR screens. What I told you in the beginning of my talk is the STK11 KEAP1 co-mutant lung adenocarcinoma may induce a unique pathway of survival, which is the pheroptosis evasion pathway. And in small cell, there is a cohort of patients who have durable benefit, but there are a minority of cases. So we now want to study the majority of cases who don't have benefit, and we're trying to find combination strategies to make them better. And I've only shown you data today about DNA damage response, but my lab also investigates. We have a completely different program which looks into epigenetic modifier. And I know there are people in the audience who are interested in epigenetic modifications and how that changes the immune microenvironment. We are also studying bispecific T cell engagers because that really brings the immune cell to the tumor cell. So we are right now starting a program that looks into that. And of course, the biology and treatment of transformed small cell is a major area of focus in the lab. With that, I would like to thank everyone in my team, and they're really wonderful people. All my collaborators at Mount Sinai, all my collaborators worldwide, they're really generous with everything. Patient samples, mouse models, cell lines. It really takes a village, and I'm truly thankful to my collaborators, patients and their families. None of this work would have been possible without the tissues that we get from them. Of course, the funding agencies and all of you for your kind attention. And I'm happy to take questions. Thank you.
Yeah. First, I think give the opportunity to the students. Thank you very much for the excellent talk. But ultimately, I have maybe last time comment because the problem remains the same because you have done excellent activity and all, but then still remains the question mark.
Yeah.
The last concluding what you said, so it is that the transformation that leads to the heterogeneity. Heterogeneity, tumor heterogeneity, particularly intratumor heterogeneity, is the biggest question. So, what you do research, what you do all permutation and combination of the treatment, ultimately everything comes to a standstill one day. And it's very soon. It's maybe 3 months, 4 months, particularly for the lung cancer.
Yeah.
It is in months, not in years. So far I have seen. So in Indian situation.
Yeah.
So that is the biggest question that we are doing a lot, but still it's praiseworthy that the way you are tackling things and excellent work. But as far as the problem is concerned as a cancer, particularly cancer like lung cancer. So that is yet there are a lot of questions to be answered.
Oh, for sure. But I think early detection, lung cancer screening, which is, more and more traction in the United States and hopefully also in India. The earlier we are able to catch this disease before they become really aggressive.
Program in India.
Yeah.
And here is my colleague, one from biotechnology, Dr. Kamal Rahul. He has a AI-based methods to actually yesterday I was so busy. We were there in the ministry. We had a more than 300 crore projects. 300 crore projects on early detection and diagnosis using AI. AI-based multi-cancer screening methods. So one of the cancer is lung cancer.
Yeah.
And 3 women cancer, cervix cancer, breast cancer, and ovarian cancer. and lung and oral cancer, because oral is very common in India and lung is a deadly cancer. So that's why, and it's a highly
So I have a question. So for the AI based method, do you use just H&E? Is H&E enough or sorry, I should be answering questions, not asking.
It's okay. It's a matter of, we should discuss out.
Yeah.
So we got a project from Pfizer and the American Society for Clinical Oncology. The project was awarded to oncologists.
Yeah.
So they approached us. So they had a data of histopathology slides. They have the PET CT scan data.
Yeah.
Then they had the genomics data and clinical data. So we built a platform or a system which looking at the radiological images,
Yeah.
we are able to do prediction at the genomic level. But your work is pretty fascinating and went into the molecular markers and other stuff.
We are doing something similar because we want to look at in H&E slides, we want to look at the tertiary lymphoid structures and how the immune microenvironment plays a role. So we are using H&E slides and the genomics data to make predictions earlier on because I think AI is going to be the future for screening because it's going to get harder and harder, and early detection is the way to go for an aggressive tumor.
I'll be happy to,
Yes, I'll be
sit
happy
down
to
and-
learn from you.
no, we both have to learn from each other. So
students, I know many of them. Unfortunately, in our group, lung cancer person is missing. We have a lot of people working on variety of major cancers, but you might remember or you might have seen the publication. One of my students did lung cancer study with the director, PhD, Sachin Singla. You must have seen his paper, but that was a clinical earlier. I don't remember. He has published so many papers that time along with me. He worked in the ICP only that time in the Noida, but his clinical guide was from AIMS director. What is his name? Guleria. Guleria that time was not director, but before that. So he was the guy, clinical collaborators guide, and I was the main guide here. So that was the one thesis only on lung cancer. But we have, huh? MicroRNA. He worked on microRNA, but he was faculty here, but then he got a chance in AIMS as a faculty in the IRCH, the Cancer Institute.
Yeah.
AIMS, so he went back to there. So at this moment, we have all types of cancer, including pancreatic and all.
So in lung cancer, the classification has been right. So in Indian scenario, you know that mutation scenario is very different compared to the
Absolutely.
Western, like KRAS and those. So the new classification, there is not any study till now which define the mutation pattern in the Indian sample. And we are doing it in the Medanta hospital
Oh, wow.
in the patient. And also we are using that data and they are doing immunotherapy. As you said that in India,
Right.
there will be no immunotherapy, but they are doing it. So when you get the patient profile, you do the immunotherapy and then we have all the data, like how it work, and then again do the sequencing and then what.
Is it published or because
No, it will get,
Oh.
it's just, so we have collected now 150 patient data. So we will be publishing it soon before anybody else publish. So it's on the way.
Wonderful.
And another thing I would like to mention that recently we are working on long non-coding RNA in the pancreatic. So we knock it down with the ASO in the mouse model, and our pathologist, that is in the Medanta, they look tumor morphology change. So I did not believe that. So I relook at the slide. So it's really changed. And now I can correlate. I learned from your presentation that there is a change
Yeah.
in the tumor phenotype. So the adenoma, the poorly differentiated change to the well differentiated. So I was very curious to know how it is happening, how I am going to defend it when we are going to write a paper. So now I get a hint that it is happening, and maybe we will talk in detail.
Absolutely.
And look at the transcription factor that are changing,
Yeah.
and maybe we are able to put it up But
I'm very happy to, because we have an entire, my lab,
mm,
almost 50% of my lab now works on cancer evolution and lineage plasticity. And we are trying to go disease agnostic, actually, because we do this in lung, because lung is my area of expertise. So I don't, but I have a postdoc who's interested in the convergence of different different cancers. So pancreatic has that, prostate has that.
prostate.
So they want to see how these different tumor histologies actually converge into becoming this neuroendocrine like very aggressive tumor. What's the bottleneck? What are the genes that actually tells you? So we are finding really interesting observations. So happy.
So happy to collaborate on the pancreatic. And you remind me my days back in NUS when I was with Kepler and we did whole exome sequencing.
Yeah.
It came. So we have done on thousands of samples
Yeah.
on leukemia, lung, and nasopharyngeal. And tumor heterogeneity also we have seen in and reported on our Nature Genetics paper. Like you cut the section into 4 pieces, you find P53 mutation in one portion, but other 3% doesn't have.
Correct.
Similarly, like you have a targetable mutation in MET in one tumor, but it doesn't have P53 mutation.
Yeah.
So,
As Sir pointed out, tumor heterogeneity is really a clinical challenge.
and this heterogeneity basically lead to the selection of the clones, which lead to the aggressive phenotype.
So the drug tolerant persisters, and they can be very different for different tumors and different therapies. And even within the same population of cells, there can be different drivers.
Yeah, so in leukemia, we were able to figure out that epigenetic modifier, which are in the complete remission. So with the leukemia, you can easily take the sample from diagnosis, remission after the treatment, and then at the relapse.
Yeah.
So we are able to figure out that epigenetic modifier are the one which persist in the uh, uh, complete remission sample. That is what we called at the morphological average. Less than 5% of the blast cell is called as complete now the complete morphological remission definition has been changed, and even now epigenetic therapy has been approved for AML. I think you know Tim Lay in WashU. Yeah. So he was our collaborator then. MLL laboratory in Munich, then Australia. So that's how we work. So I think I pretty much enjoyed because I can see what I have done and how it is going.
Thank you.
Thank
Thank
you.
you.
Thank you.
Yeah, it's great. Actually, the same thing. Actually, we cannot conclude anything in short, but it's very interesting because the whole world is interested on the heterogenic population in the cancer.
Yeah.
So it's great that is if you make kind of classification on that. And one guy came in our institute also from London. Who is his name actually in the pancreatic cancer? He was showing not only in the cancer population in the some of the cancer associate cells also showing similar kind of the transcriptome profile.
Yeah.
So it's very hard to believe, but even very challenging to identify the biomarker for that. So
Right.
It's great. So you must be doing the classification of the subpopulation of the small.
Right. So
yeah.
I did not show anything about that, but because we have now transcriptomically classified small cell into like 4 major subtypes.
Yes.
ACL1, Neuro-T1, PAU2F3, YAP1, BC plasticity within subtypes. So yeah, that's a completely different story. I can
Yeah,
go on.
even it come with the other cancer also. The origin can be
Yes.
in the
yes.
in this
The cell of
similar
origin
cell
can be
of
different. Yeah. Yeah.
Great. Anyway, I
Thank
thought
you.
that,
Thank
yeah,
you.
yeah. I'm expecting the questions from the. Yeah,
There
go
are no
ahead.
stupid questions.
Hello, ma'am.
Hi.
Like your talk, but
you.
in one of your study, you have shown that when you have done that anti-PDL1 and in combination of other compound, there is a significant rise in the effector memory cells. So it's not possible that there may be some changes in the central memory cells, or may there be the number rise in the marrow infiltrating lymphocytes and tumor infiltrating, because now the in immune suppression mostly people are targeting marrow infiltrating lymphocytes and tumor infiltrating.
Right. So we haven't looked at that's a great question, but we haven't looked into that. We only resected the tumor and looked into just the tumor. We haven't looked into that. What we predict is happening. We are getting upregulation of the CD8 positive T cells, right? But we have also tested that these are not just upregulation, these are not just increase in number, these are actually functional because we have looked at CD69, we've looked at interferon gamma, and we have done in the cell culture cytolysis, T cell mediated co cultures to see that these are actually functional T cells. We get memory affected T cell population higher, and what we predict that is happening is it is making the tumor sustained. So in one of my slides, and I don't know if I showed it, that these are not just 20 days of response, these are 4 months, 5 months of tumor regression. So, which means it is actually changing the entire tumor in itself so that it's a sustained anti tumor effect that we get. And that's why we think the CD62L and 44 positive cells, they are contributing to the sustained anti tumor response that we get. But we haven't looked into the marrow infiltration and all of that.
Yeah,
It's a great
because
question.
if rise in the factor, there is the higher chance that there is an improvement in the tumor infiltrating lymphocytes if we look in that tumor microenvironment.
Right, right.
Yeah.
And I think the more we look into clinical samples in the single cell level, all these questions can be answered a lot more actually confidently.
Okay, thank
Thank
you,
you.
ma'am.
Thank you. Yeah, sure. Yeah, great question. So we think that when you have a KRAS mutant tumor in combination with an STK11 loss and a KEAP1 loss, so with what keep-on loss does is it changes the NRF2 axis, right? And NRF2 can transcriptionally upregulate SCD1. So we think it is going through the NRF2 axis and it is changing the SCD1 levels. It's a great question, but we think it's the NRF2 axis that's upregulating SCD1. And SCD1 is changing the fatty acid metabolism, and that's leading to the ferroptosis. But I think it's through the NRF2 axis that SCD1 is changing. They're active. So it's a loss of function mutation, which means the function of STK11 is lost. It's a tumor suppressor. So it's a loss of tumor suppressor. Yeah. Yeah. Sure. Correct. 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 Correct.
So when you were doing this inhibition of GPX4, at that time I wanted to ask you whether it's happening because NRF2 is getting affected because of the people mutation, or it is just solely the GPX4 which should occur in the antioxidant pathway.
Great question. So the GPX4, we treated GPX4, it is a master regulator of ferroptosis, right? So we wanted to see the reason we did GPX4 is we wanted to see whether these cells were actually responding to a ferroptosis targeted therapy. We don't see STK11 or KEAP1 directly regulating GPX4. So when we looked into the clinical sample, the GPX levels did not change. What changed was the SLC7A11. So it's not through because GPX4 it controls the glutathione pathway, and SLC7A11 it controls the cysteine methionine.
Now I clearly got it. It's going through the different route rather than this route. Okay.
It is not changing the glutathione; it's changing the cysteine.
And because of the cysteine pump, it is getting
correct.
into.
Correct. Hi.
Heterogenic.
So, ma'am, in the second study where you identified P53 loss of function and RB loss of function,
I did not identify. It was a long before my time. It was an 1985 study.
Yeah. So, like, did you people try to look at its epigenetic regulation? As you mentioned that you
Oh,
people
yeah.
are actually working on epigenetic modifiers. So, did you people like went for its transcriptional and epigenetic regulation to look into it? Like, can we target from that point? Or
Oh,
not?
yeah, we have a study. I did not, of course, I did not show those papers. So since you asked, so what small cell lung cancer does, it is not a mutationally driven cancer, right? So if in a cancer where everything is mutated, nothing is mutated because mutation doesn't matter at that point. It's a high tumor mutation burden. So when we looked at the transcriptional level, it's a very transcriptionally addicted cancer. So it is addicted to its transcription factors. And epigenetic modifiers completely change. So what we studied is that the MHC class 1 suppression that you see, it's actually epigenetically controlled, which means the EZH2 family, like the PRC2 complex that you see, EZH2 transcriptionally represses MHC class 1 in small cell lung cancer. So when we de-repress, when we target these cancers either with an EZH2 inhibitor or with an LSD1 inhibitor, so changing the epigenetic machinery, we were able to upregulate MHC class 1. So we were able to take away the immune suppression that we see. So epigenetics is very important in small cell. I did not show you, but we have an entire study that we published on LSD1 inhibition and how it de-represses MHC class 1 in small cell. It's a very good question.
Thank you. Thank you. Ma'am, as you were talking about the diagnostic point of view, so like, are you people trying to develop some kind of biomarkers which can be given to the like smokers for like?
So you're asking
early
me
detection?
exactly about the slides I took out. So now I'm like, okay, I should have put those slides in. You're talking about the slide. So biomarker is a huge debate in small cell because there are no biomarkers. It's a heavily mutated disease. So when the patient comes into the clinic, everyone gets chemotherapy, immunotherapy. There is no biomarker. Everyone, it's a one size fits all approach that is given, right? So we are actually looking into blood because it's a very aggressive tumor, which means there's a lot of shedding of tumor cells in the blood. Tissue is a problem. So now we are trying to develop blood-based biomarkers, which means we take out blood, we look into either the methylation profiling in the blood or the CTCs, the circulating tumor cells, cfDNA profiling, and we're trying to detect biomarkers in the blood, no longer in the tissue because it's hard to get. So we are trying to develop, we are not there yet. We have few indications, but we are not ready for clinical prime time. We are not even there to do biomarker based. We are doing a biomarker based trial with Schlafen 11. Again, it's a completely different story that I did not have time to get into. Biomarker is a completely different area. But we have very slim pickings when it comes to biomarker, and I think blood based biomarkers will be the future in this aggressive tumor.
But as it is aggressive and it's already set down, isn't it like it will be at very later stage there?
Yeah, so that's why cancer screening is so important, because when we do cancer screening, we will not just catch lung adeno, we will also catch small cell, right? Because we will catch everything. So if we are able to detect it really early at the limited stage, in the limited stage setting, small cell is confined to just the lung.
Yeah.
In the extensive stage setting, it goes from lung to the liver to the brain. So then it is very hard. So if you're able to do early screening, if you're able to detect them at a limited stage setting where they're still localized in the lung, then we can actually do something about it. In terms of biomarkers, as I said, we don't have very confidence about the biomarkers that we currently have. But I think as we look into more tissues, look into more blood samples, we will be able to get biomarkers that we are more confident about.
And now I'll ask the stupid question.
There's no stupid question.
So like, like, because I'm not a lung cancer person, I'm working on ovarian cancer, so I don't know much about it. So like, if there is some screening which can correlate it with the upper respiratory tract from like trachea and like, so that, because biopsies will be hard for taken from lung, but like if it is correlated to like the upper respiratory tract, and from there if we can do some screening in smokers, so maybe it will.
So the smoking guidelines is usually they should be up after a certain age, right? So it's usually 50 up. Patients who have a long smoking history, they are eligible for lung cancer screening and insurance covers it. I don't know how it is in India, but that's the rule in the United States. They need to be at a certain age limit and they need to have a long smoking history and they are eligible for screening. Though we have recently published a Lancet paper where we are saying that it should be relaxed because genetic determinants, there are germline determinants of lung cancer that we don't talk about. So there should be, screening should be open for all. Like irrespective of their smoking history, because women are getting lung cancer at a much, much early age. And women lung cancers, they happen in never smokers. So smoking is no longer deciding whether a person is getting lung cancer or not. It's happening in never smoker women. They're getting EGFR mutant lung tumor. So we are relaxing that. But COPD patients, so when they go into screening, if we are able to detect it early, if we are able to have a confidence biomarker that they will ultimately develop lung cancer, that will be very important. But I think screening will help.
Thank you.
Thank you. Thank you.
I think one thing I want to comment, what you just now commented, that mutations and all is not helping, but it is not for the small cell lung cancer, but non-small cell lung
It
cancer.
is very helpful.
Very helpful.
Very
Just
helpful.
ALK, that our colleague, I was coordinating everything about him, his treatment and all, and doctors and all. So he had the ALK mutation,
Yeah.
and he survived for 3 years, more than 3
Oh,
years.
now there is a new Crown study that came out. It is showing 7-year survival with ALK mutation on alectinib. So non-small cell has really benefited from these targeted therapies that are mutation driven.
Yeah.
Oh, very important.
That is very helpful. Yeah.
Yeah. No, osimertinib, there are 15, 20-year survivors now on osimertinib. So if they work, they really work.
It ultimately developed the resistance.
Yeah.
And that is the major thing. But resistance is not because of the heterogeneity. Heterogeneity is there, but there may be some other factors.
Pre-existing clones, there may be
There
other.
are stem cells and
Correct.
the other cells.
Correct.
Then the other, what is that cells? The other cells, no, the stromal cells. Stromal cells, they also cause the resistance.
Correct.
The drug resistance. So there are many other factors, including cancer stem cell, but heterogeneity, I think 20 years back I observed, but it is not yet published, that in breast cancer we find that the one gene, I was working on AP1,
Mhm.
that,
Yes.
that the FRA1 is one place in the same stage of breast cancer. Well, even in the same breast cancer, different site, it is the FRA1 is overexpressed, acts like oncogene, and somewhere it is tumor suppressor. Even between the patients and within same patients in the different tumor sites.
Yeah.
And that is yet I need to have more data to publish that. But then
Yeah.
but I proposed and I told in many papers that there is a heterogeneity. Means intratumoral heterogeneity.
Correct.
Yeah. So it is not that you do a marker and then it is all right. And you do a molecular or histological is a little different, HNSC, but there also immunohistochemistry you do. And the pathologists do not do the whole site screening of the whole
Right.
slides. So you may leave somewhere which is majoritarily you find some thing is expressing and you take it as a positive.
And also from where the slide has been taken, it's not the whole tumor, right? They cut a section.
So that's
correct. So with AI, now I think the next thing is, can we predict even the part that is not there? Can we train machine learning in a way that we have a certain section, but can we train it enough to predict
that is
the area that is not there in
for
the slide?
example, service cancer now.
Yeah.
One paper, one of our colleagues have published, Datta. Yesterday he was showing to the minister. So it is, of course, there are a lot more things to be done. He has added all the clinical epidemiological informations, their age, and the particularly for breast cancer. So he was telling breast cancer, no? Service cancer, mainly service cancer. That he has all the clinical epidemiological factors, few important factors, the age of marriage and other things, and the histological and the biomarkers informations. Put together, you can predict whether what type of progression or you will have the what type of
The risk of
developing cervical cancer. So that type can be done in the also for lung cancer. But the what is you said, and everybody now feels that AI is going to change the everything. So, but AI, it is all like the atomic energy. It has a bad side of it. So AI has a lot of bad side, but at least for the disease diagnosis and prediction, it will be great help if AI is used
To take away human error, fatigue,
but
like,
the
you know,
problem is
neurologists are
that
really
I
busy.
do not know in US as well as in India, there is it's a new discipline now. So it is a completely you need to have a trained manpower.
Yeah.
The lack of manpower is there. So you need to have because those who are now getting trained, there are only few coming out, and there are everyday change is coming in the AI field.
Right.
So it will take few years to get stabilized, and like it came the IT time. So like that, AI will again transform into AI. It will take time.
Yeah, yeah.
Okay. Any other comments? Anybody?
So I had a question. Like you have in your lab, have you developed such models, prediction models, any of such?
So we
For
are actually
impacts?
collaborating with an AI people to do. Are you talking about AI predictions?
Yeah.
Yeah. So we're collaborating with a company. We don't have in-house prediction models because we don't do that. But we are collaborating with someone, and I'll be happy to collaborate with anyone who will teach me about predictions with AI. So, you know, I would be.
Okay.
Are you working on that?
Yeah, we have written the joint project with Sir was our main lead.
Okay.
And the project with Sir was discussing. He majorly work in
Correct.
Regional health paper on this model, and we are in the process of scaling up and talking to other hospitals.
Wonderful, wonderful. I would love to, as I said, I mean, I'm just entering the field because it's a need almost of doing that. And I'll, you know, I would love to keep in touch and then learn from you guys or collaborate on how we can use the slides that we have. And hopefully you guys can lead the effort. I mean, I'll be very happy to collaborate. Correct. Right. Getting the right yeah, correct. Yeah, 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 yeah.
that's sir 5 years ahead of yours.
Yes.
So that is the great case.
That is the dream, right? That is the dream. Yeah. Like if I can detect 5 years ahead, there's so much we can do. Yeah, 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 yeah. I need to.
Yes. I need a photo with all of you, but I'll give my phone to you also so that I can.
No, no, we'll send the photos.
on WhatsApp.
Yeah.
Take Lily knows.
Oh, okay, okay.
I'll share this. You share? Maybe. I mean, after.
But it's really great that in a small period of time you are able to integrate many clinical jobs.
Thank you. Thank you. I have wonderful colleagues. Wonderful
सारे आ रहे हैं? Yes. Yes.
Yeah.
Yeah, I really like that.
I love it. Yeah. Thank you. Thank you.
Good job, but look forward for collaboration.
Yes, yes, yes.
And